All right, we are live. I forgot to stream it on Monday. Didn't even think about it since we were just up on the board. Couple things. IDEA survey sent out an announcement. I've updated. There's it's going to be our our breakdown. So based on percentages, percentage response rate, that would be the percent that's added to the overall course grade. So where do we stand right now? We are at 65%. So not quite to 90. 17 enrolled students, 65%. So what, nine students maybe, 10 students, 10 of you. So we can go ahead and do that. Also, got the exams, got the practicals graded. I went ahead and have the grades posted on Blackboard. So for the exam, you can go to the grade scope to see the multiple choice, multiple answer section. I've posted the keys outside my office, so you can, along with a copy of the exam, definitely grab it. But then I also have your current course grade, your current lecture grade, and then your current lecture, current lab, and then current course grade. So that kind of helps you estimate where you are at. Again, we have the blood drive. It's on Thursday, so donate blood, get someone to donate for you. Try to go and get turned down. That'll get added to exam number 3. The scenario that we have, that is due when you come to take the exam, the final exam. That's up to 5 points on exam number 2. Both of those exams are 25% of the lecture grade, so those are pretty big percentages. So each of those exams had about 100 points, I think 98 points, and then 100 points for exam 3. So getting 5 points added could be significant. Also, remember the final exam, I'll replace the lowest exam grade with the final exam grade if the final exam grade is higher. I do check on exam number 1 and number 2. So exam 1 is only worth 20%, exam 2 is 25%. If you do better, and your exam 1 and 2 grades were similar, but let's say exam 1 is lower by, let's say, a point, or let's say 2%, what I'll go in is I'll manually make the replacement of exam 1 and exam 2 and do whatever one helps the most. So sometimes it's exam 2, it just kind of depends on how close they are, and as well as how you did on that final. How much better did you do on the final? Final exam, 10 a.m. Monday. Working on it. It'll be basically the same format. Multiple answer, multiple choice section. It's only one answer for those. You'll have disease names. So you'll get the word list of the parasite species. I give you the disease name. You give me the parasite species. We do have a lot of species that we covered, so it's going to be pretty good. Pretty good section. There's a life cycle section. The life cycles will be kind of like what we did on this 3rd exam, where I'll have the life cycle drawn out with letters or blanks or whatever, and ask you questions about that life cycle. We do have quite a few life cycles, so might check it out. Might start reviewing it. The symptoms, we do have a lot, and if you go through and start looking up like the diagnosis section, you'll see that we have a lot of overlap in a lot of the symptoms. We could get by with the diagnosis section on the individual exams because, like all the nematodes, the different species of nematodes had different some different specifics. That's not going to be the case once we start including the platyhelminths, the nematodes, well, not really the acanthocephalans, but all of the protozoans. So we're not really going to have more of a diagnosis section. We'll probably just have a section where I ask about some symptoms of some of the infections, some of the diseases. I'm trying to decide how we're going to do that. If that'll be like a selection, you pick, you mark which of the symptoms apply, or if it's just freehand, like list the symptoms. Will say at this point, go back to the first lectures where we talked about the overview for the class and the common themes. So go back to those common themes and think about how they apply throughout all of our parasites. We've been doing a lot of the same things. We've been talking about adaptations for successful transmission. We've been talking about immune evasion mechanisms. We've been talking about pathology aspects of the parasites. So kind of go back and start looking at, review those common themes, and then pick out examples of just across all of the parasites that we've covered. It is going to be a longer exam, so prep for it. Any questions? No? All right, so I think I have everything. All right, let's we talked about aggregation, clumping of parasites. Basically, this is dispersion patterns. It's very characteristic of parasites in general. It is more rare to have something not aggregated into a host. So what causes it? So we have types of dispersion. Is this number 2? We have pattern. Number 1 is patterns. All right, number 2. Causes of aggregation. How do we get that? As I said, this is the most common pattern. Variance in mean ratio is greater than 1 because you've got a lot of variation compared to the average number of worms. What causes that? A couple different things. Primary one is heterogeneous exposure. So hosts are going to be exposed differently to these stages. All of this is basically related to parasite recruitment or the accumulation of parasites. So that's what we're talking about here. Heterogeneity of exposure to the infective stage. What's going to generate that? First thing is host traits. Host traits can produce this difference. So we can use example size of the host to show that larger hosts need more energy They need more energy. So what do? They feed more. All right, and this is like twofold. One, they're consuming more food, or they're consuming bigger food items. More food exposes you to more parasites, as we talked about intermediate hosts having those larval stages. Larger hosts, larger prey items are probably consuming some of the smaller intermediate hosts. So the larger prey item is going theory, have more parasites. But also, in this desire to feed more and acquire more energy, they're going to be moving around in their habitat much more. And if you're moving around, you're more likely to encounter these parasite stages and thus become infected more. So larger hosts need more energy, so they feed more. If we were to look just at host size and do either prevalence, or we can talk about intensity, either one, we see this positive relationship. All right? So host traits, host size is 1. But we also have age and sex differences. So size differences definitely play a role. Sometimes size isn't necessarily correlated with age. Sometimes it is. Older individuals tend to be larger. But with these age and sex differences, what we're talking about variation in foraging, in diet, and so forth. So differences in foraging strategy, diets, and social behavior. Probably learn this in some other classes, maybe. But females, especially going into the reproductive season, are going to start shifting their diet to try to get more high energy food items relative to the male. And those high energy food items may be more or less effective. That also would expose them, for those females, to a different set of parasites. Also, if we talk about social behavior, you have these animals that will form herds, and sometimes those herds are just basically female herds along with the young. They're going to be exposed to a different group of parasites than the males, who might be more isolationist in nature. So host traits can lead to this difference of exposure, and that would produce some of our aggregated patterns. Second thing, with this heterogeneity of exposure, is related to the distribution of infective stages. Distribution of infection. All right. And this is kind of twofold. So we also, when we think about infective stages, we have to think about how those are distributed. So if we're looking at adult parasites, then we need to look at the distribution of our parasites in that intermediate host. And in many cases, those in that intermediate host, the larval stages will also be cloned. So some intermediate hosts are heavily infected, right, but most of them are not. If they're not using the intermediate host, then we also have this distribution of the free living stages. So on these, you can think of dung piles. So if our intermediate, or if our definitive host is, let's say, a cow, and it has, let's say, a beef tapeworm, which is what species, by the way? Common names. I should also say, common names that are also on the exam. So, Tenia. Which one? Saginata. All right. So if Saginata is infected and it defecates, leaves this dung pile, you're going to have a bunch of eggs, a bunch of eggs coming out in that dung pile and be just in that localized area. All right. You go 10 feet away from that, there's no dung pile, there's no parasites. So if something comes along to feed on that dung pile, they're going to be hit with a lot of different infective stages versus something else that comes 10 feet away to feed and so forth. So we've got distribution of the infective stages and then host traits. That leads to this heterogeneity of exposure. So all of this is basically non-random aggregations. And that's kind of a key point. So non-random aggregations of larval stages, any larval stages, non-random aggregations of larval stages are magnified at higher host levels. So if our distribution pattern is aggregated or clumped at the egg stage, then that is likely leads to clumping at our intermediate host stage and clumping at our definitive host stage. It just kind of magnifies up from there. So heterogeneity of host exposure. The other thing that can lead to this clumping or aggregation is variation in host susceptibility. There is a genetic basis for resistance or susceptibility. Some individuals may be more likely to harbor an infection, or some individuals may be more likely to harbor a heavy infection and still survive. We have examples of these throughout all the parasites. Not all parasite species are going to exhibit this. Not all individuals are going to be resistant or susceptible to parasites. But it is a factor that can lead to this aggregation. All right. So we talked about the causes. Let's talk about the consequences. And we've already stated some. So we talked about the effect on the host. We mentioned the effect on the parasite as well. But the effect on the host is basically death, so death will be more likely in our heavily infected host. But the other thing is related to evolution. So heavy infections are a stronger selective force for resistance. And maybe resistance isn't the best. Oh, yeah. Worst thing is when you leave the alarm and you're gone. And your wife wakes up at 6 in the morning because my alarm's going off. I'm not even there. Oops, sorry. All right, so resistance, yeah, that is the selection pressure for resistance, but if we think about evolutionary history, these parasites really had a strong selective force for the immune system. So our immune system, the complexity of our immune system was basically, or at least the hypothesis, is basically driven by the parasites, and you didn't really get to that level until you started seeing vertebrate evolution. Once the vertebrates came around and you started having these larger predators, now you start getting more and more of these parasites, which apply a much stronger selective pressure to develop a host immune system to try to eliminate those parasites. So the effect on the host, the heavily infected individuals are going to be more likely to die, and that thus gives a very strong selective pressure for evolution of resistance. What about for the parasite population? All right, effect on the parasite. You can say, we mentioned this, heavily infected hosts are more likely to die. And thus they take out a large number of parasites. So in a lot of ways, that's kind of bad for the parasite. You take out a huge number. That's part of the aggregation, is that a small number of hosts harbor most of the parasites in your system. If even one of those gets killed, you're taking out a large number, which could disrupt transmission. I say could because it's probably there. You probably need several of these heavily infected individuals to get taken out in order to reduce our net reproductive rate below that 1. So heavily infected hosts are more likely to die and take out a large number of parasites. And then the other thing, part of this, is this idea that the parasites are basically putting all their eggs in one basket. By being aggregated, you've placed all of your transmission stages, perhaps. We'll use transmission stages. Let's say 4 larval stages. You've put all these larval stages into a very small number of hosts, and you're relying on those small number of hosts to be consumed by the definitive host. All right? You can say there's 100 prey items, but only 3 of them have a lot of these parasites. Well, if those 3 don't get eaten, again, those are a lot of larval stages that never make it. Those are a lot of larval stages that never make it. So, in essence, the parasite has kind of invested, or at least the pattern of transmission, the pattern of the environment, and all the behaviors have led to this situation where a lot of their transmission stages are in a few individuals, and you're relying on some of those to actually get consumed to complete our life cycle. It's not necessarily a major problem because these parasites have evolved and they still exist. So that leads us then to theoretical treatment options utilizing this idea that most of these parasites are aggregated within the host population. So our theoretical treatment options. So what we're going to do is say, imagine a population. So each of these boxes represents an individual in our population. And the size of the boxes represent basically the intensity of the infection in our host. Okay. And we could say we have a bunch of uninfected individuals too, and those are going to be circles. So the circle, uninfected. Our squares represent infected. to scatter our uninfected around our population. So we're aggregated. Most of our parasites occur in a very small number of our hosts. And most of our hosts have a small number of worms. Right? So in this case, we go out to a population, we see the parasites. Our parasite likely has a net reproductive rate of about 1. And why do I say that? Well, I say that because when we actually look at parasites in host populations, you generally don't see drastic changes from year to year. You see variation in prevalence, you see variation in intensity related to season and just stochastic events, but for the most part, if we were to survey it 10 years right in a row, we're going to be about the same with our averages. That means our parasite population is neither increasing or decreasing. It's basically in equilibrium, you could say, or it's at least stabilized. So our parasite population likely are approximately equal to 1. All right? So what we want to do is treat our individuals to get a net reproductive rate below 1. That's our goal. Why is that our goal? Yep. Over time, parasite goes locally extinct. All right. So how do we start to approach this? What can we do? What are our options to try to get to this point? What's that? How? So one way is to block transmission. Right? If we can prevent any of these new individuals from becoming infected, right, then we're basically putting up a stopgap to get to this point. Our example, exactly. Apolysis, or what was the common name? Anywhere. Good review. And as I said, I think for studying, I would be going through my notes and writing down parasite name, and then your common name, and then the disease name that goes with it, or disease names that go with it, and then possibly some symptoms with each of those, and just kind of start assembling. All right, so block transmission example, Dracunculus medinensis, and they did that. What has to happen, though, for this blocking of the transmission to really work and eliminate our parasite in a short amount of time? Yeah, we can't have other things getting infected and dumping the transmission stages back into the environment. What else? What else? Would this work for schistosoma? What do you think? You're shaking your head. Why not? Okay, so the way What if we could develop a cream that would stop that from happening? Sci-fi. Well, okay, so we did. We blocked it. Would it be successful? Eventually. But how long would we have to wait? We're waiting a long time. For, let's say diphyllobothrium, that was 1 year. Adult worm gets into the host, matures, dumps its eggs, it's died. And that definitive book says it has to get reinfected every year. With schistosoma, that worm's going to live 20, 30 years maybe. So you've got a long time to treat for this blocking the transmission to have any success. This Dracunculus really wasn't going for very long. Yeah, you've got about a year or so. So you said 8 to 10 months from when you get ingested to when that blister starts to form. You block transmission, you can treat it over the course of several years, and you should see immediate responses. You might not see it with longer-lived species. So yeah, blocking transmission, that's one option. What's another option? What's that? I don't think that's going to be possible. I mean, that's kind of what some of the drugs do.
Is it treatment?
What's that?
Medication and treatment.
Of who?
Killing the parasites inside your body.
Yeah. All of them? So yeah, we can treat all individuals. That's a possibility. So we just go into this population, just give everyone drugs. And we do that, right? We do that. You give drugs to you go into some of these villages that have schistosomiasis, just give it to everyone. Is that a good thing? No, why not? Sorry?
Do they gain resistance to the drug?
Okay. That's actually, we have a case study for malaria. I believe it was India treated all of their salt with an anti-malarial. And within a year, they started saying the drug-resistant malaria. Right? So treating all of them. What other things?
Instead of individuals, like the environment.
I would put that in the block transmission part, or blocking the environment. So at least for us, focusing on the human population. So, but treating all the individuals, you're right. Well, it's a good thing in the sense that we're not excluding people, but it's a bad thing because it could lead to resistance. Could also be a logistic nightmare. And also a monetary nightmare. You're treating everyone. That's a lot of money. You have to make sure that everyone gets treated. Right? And everyone actually follows through on taking the treatment. So maybe that's not going to be the best way. And if we're not going to treat everyone, then perhaps we could target heavily infected individuals. We go through, treat those, and then by treating those, we've drastically reduced the parasite population, which means we have fewer infective stages going out into the environment, which thus has the potential of reducing this. Again, this also has issues. It's got ethical concerns. Who has filled out an IRB protocol? Why'd you have, what is IRB? Institutional Review Board, right? All right, why did you have to do that?
Well, my research involved
Yeah, so you had to fill it out because you had a pathogen that could infect humans, right? And you're studying parasites that could infect humans or actually would infect humans. You need to get approval to go in to say, yeah, we're going to go do this. And you're also at the human research board. That's also, some institutions have them separate, so you've got like your biosafety committee and then like the humans and research committee or whatever, but you need approval. Do you think some people are going to like it when you say, yeah, we go into this population where 80% of the individuals have a parasite, but we're only going to go treat 10% of them because those are the 10% that are heavily infected. Do you think they're going to be happy about that? Not at all. Not at all. They're going to be that kind of gets the ethical issue, and that has kind of gotten into the way of this. So mathematically, we could demonstrate that this may be the best way, best use of funds. So it's going to be cheaper. It's going to be a little bit better logistically because you have fewer number of individuals to actually follow to make sure that they do continue their treatments and everything. And we've got that, and some studies have actually done this sort of thing, but institutional review boards have always said, okay, how long is this going to last? We're going to give you a year, and at the end of the year, you then have to go in and treat everyone. That fits your ethical concerns, and it's like, okay, well, we treated them, but parasite still comes back because you had all these other things that are out there, including larval stages in the environment, reservoir hosts, and all that other stuff. But this seems to be the way in which a lot of the control mechanisms are going. It's a good way because we're not promoting resistance in any way. There is that selective pressure, but we're not causing blanket resistance. So those are our treatment options, and again, if you're interested in more of this, there's quite a few review articles that look at different ways to treat and the pros and cons and everything. But this is how we can use aggregation to our benefit. You can say it to our benefit. It's still parasites. All right, questions? Move on. Get through this. We will. So we're going to go back to that equation. So we had the equation that described the change in population size. We had aggregation. Aggregation's a big part of parasites, but so is transmission rates. All right. So what affects transmission rates? Number one is host population size. Host population size. So with our host population size, increased number of susceptible individuals usually leads to increased chance of transmission. We are more likely that the parasite will encounter a host that they can actually infect. So hopefully that's self-explanatory. However, we also have something called the dilution effect. So if we start having a large number of resistance, we'll actually have a decreased chance of transmission. Decreased chance of transmission, and it's actually a much larger decreased chance of transmission than what you would predict. Because now what you're having is these hosts, these resistant hosts, still picking up the parasites at the usual rate, But those guys present a dead-end path for the parasite. None of those parasites are going to reproduce and spread it out. So, our R0 is going to decline, prevalence is going to decline, all of this. We see this anytime you have, let's say, an introduction into an area, let's say, an invasive species that does not serve as a host for the parasites in that system. They can acquire it. Oftentimes, you'll start seeing a decrease in your prevalences in the host population because of this effect. You basically diluted the number of available hosts in your population. And that's due to resistance. So also that would apply to immune individuals as well, not just resistance. So you can kind of go back to immunity versus resistance, those ideas. Reproductive rate. And this was at lambda. Right. So if we increase output, then we're going to increase transmission. And we'll give just a couple examples so we can kind of get to these last two. All right, I'll leave the rest of these examples to you as you study for your final exam. But asexual reproduction is one. We have that in our digenetic trematodes, where the redi or the spore cysts reproduce asexually, increase in number of reproductive stages, I should say infective stages, increase the number of cercaria that enter our environment. So one egg from that adult is going to produce thousands upon thousands of these cercaria. That's all about increasing reproductive output. We'll give one more. Apolysis. What's apolysis? Remember that? Those egg sacs. What are those called? Proglottids. Yeah, proglottids drop off and go out to the environment. And in some cases, this proglottid will drop before it's completely mature. I should say before it's completely gravid. So by dropping it off, it gives more room for that worm to increase its strobilization and produce more and more proglottids. So in a way, you're producing them and you're dropping them off before they're fully formed, before they're fully gravid, because these will get gravid on their way out. This is a way to increase output. And then there's several more that we talked about, and you can have examples. Have a species that lists an example of how they increase their reproductive output. All right, so reproductive rate, it's all about increasing our output. Number 3, I should also say one other one. Lifespan would also fit into that. So we've mentioned it a couple times. Long-lived hosts, release more eggs over the life compared to some of these short-lived individuals. However, those short-lived individuals have other adaptations that might increase their reproductive output. Death of our transmission stages. That's mu in our equation. So if you decrease death then we decrease or increase our transmission. So this is kind of a reciprocal inverse relationship. Increase death rates, you decrease transmission. You decrease death rates, you increase transmission. This has led to the evolution of resting stages, . So your metacercaria, any of those oocysts that enter the environment, they can persist for a long time. They have a very low mortality rate. And by decreasing your mortality rate, you basically lengthen that transmission window. Don't forget our eggs, especially if they're resistant eggs, all kind of fall under these rusting stages. But we also have limited activity. Of the stages. And it could be behavioral. So these are not insisted. But maybe they just sit there not moving until they receive some sort of cue. And the cue could be environmental. So maybe they just sit there and rest until they start sensing a decrease in salinity or a decrease in temperature, but that might mean an influx of fresh water following the rain. And then all of a sudden they say, okay, conditions are going to be good for transmission for the hosts are going to be around, let me now start to increase my activity. Both of these cases, we're doing things that decrease our energy usage while we're in that transmission stage, because our transmission stages are not feeding. They're relying on that stored energy. Questions on that? Our last one was transmission efficiency. Transmission efficiencies are all of those other things that we had talked about. So transmission efficiency was when you did encounter, how likely are you to actually transmit, or it's a modifier that changes your interaction rate. So you're talking about behavioral responses. Behavioral alterations. And morphological adaptations. So your behavioral responses would be all of the taxis behaviors. So moving towards light, moving away from gravity. For hookworms, moving up towards the surface when they're ready to infect. All right. So those are all the behavioral responses that will change the transmission efficiency of our parasites. Our behavioral alterations are all of those things that increase predation rates. And we've had a lot of these. Right? I'm just going to put fearless mice because that was one of the most recent ones that we talked about, right? But we also had the parasites themselves, right? Increased feeding of mosquitoes when they're infected with Plasmodium. And our morphological adaptations, we haven't really had a whole lot of these. But there are examples where the transmission stages look like a food item. So the eggs of some of the cestodes, they look like the food item of the aquatic crustaceans. And that's going to increase your transmission efficiency because instead of having a random contact between parasite and host, you're now going to start having this non-random movement that's going to increase our transmission. All right.
So these are kind of like we talk about all of our adaptations to increase transmission. I'm going to leave this to you as you go through and study your life cycles to find these and say, oh yeah, this adaptation might influence the reproductive rate, or it might influence death, and transmission efficiency, and so forth. All right. All right, so I'm going to pass back these exams. They are in alphabetical order. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We're missing some people. They can come get them. It's going to be tough. I've got meetings today, and I